DNA, the universal code of life, can be altered to make useful medical and scientific materials, according to a study from the Scripps Research Institute. Moreover, the process isn't just a laboratory experiment, but can be put to use practically. The modified form of DNA is more stable than natural DNA and possesses additional desirable properties, according to the study by Floyd Romsberg and Tingji Chen, a scientist in Romsberg's lab. Research into these properties is ongoing. For example, the modified DNA could be used to make a hydrogel, a water-containing substance that can deliver drugs, or shaped into nano-sized structures, or nanomaterials. In addition, the synthetic DNA could itself be made into a drug, surviving in the body when natural DNA outside a cell would be quickly destroyed. Biology is full of enzymes called DNAses and RNases, and these are enzymes that specifically degrade RNA and DNA, Romsberg said. And it's been a real limitation of the field of trying to use DNA for any purpose in medicine as a therapeutic. But those enzymes don't cleave the synthetic DNA. The study was published October 10 in the journal Angewandte Chemie. It can be found at j.m. Romsberg. Moreover, the modified DNA can be precisely replicated, using an enzyme Chen developed for that purpose. That accomplishment was detailed in a study published last year in Nature Chemistry. It can be found at j.m. ppcr. Evolving function Chen produced the enzyme through a process of directed evolution, selecting for enzyme variants that were steadily more productive in replicating the modified DNA. The artificial enzyme's role is reminiscent of the development of polymerase chain reaction, or PCR, a workhorse technology that's a foundation of biotechnology. It uses a natural enzyme that replicates DNA, necessary for cells to divide and life forms to grow. Like its natural equivalent, the synthetic enzyme replicates modified DNA automatically. That means an engineered DNA molecule can be mass-produced in the manner that natural DNA is now manufactured, Romsberg said. That combination of precise engineering, stability and ease of production of nanomaterials gives the modified DNA great power, Romsberg said. Romsberg has explored the use of DNA for decades. In 2014, a team he led produced the first life form that incorporates an expanded DNA alphabet. Instead of DNA's natural 4 letters, the As, Ts, Cs and Gs, the engineered bacterium can use 6, the additional 2 being called X and Y. The 2 extra letters can give the organism properties impossible to produce with natural biology. That work continues in Romsberg's lab, and at Synthorx, a San Diego biotech company that licensed the technology from the Scripps Research Institute. Meanwhile, Romsberg has continued on a parallel path to investigate the use of DNA outside cells. DNA is in many ways an ideal polymer, capable of being designed with exact specificity, Romsberg said. Polymers are materials made up of simpler building blocks added together. Rubber, wool, silk and synthetic plastics are common polymers. Most people don't think of DNA as a polymer, Romsberg said. But it actually solves several of the problems that the polymer field has. Producing polymers of controlled length is really hard with normal, conventional polymers. And the exact composition of the polymer is also really very difficult to make. Precise engineering provides precise control over length and composition, he said. Its letters are arranged in a sequence that spells out the genetic code. And mutant DNA polymerase, the enzyme Chen evolved, duplicates DNA strands. That control provides the ability to arrange the DNA letters, or nucleotides, in exactly specified locations, and to create three-dimensional structures at a molecular scale, useful for creating nanomaterials. Besides introducing automatic replication of modified DNA, the last year's Nature Chemistry study described some modifications to the DNA letters to improve stability, such as the attachment of fluorine atoms at specific places. The new study added molecular hooks that can grab onto other molecules. These hooks operate through a process called click chemistry, pioneered at the Scripps Research Institute by Nobel laureate K. Barry Sharpless. As its name implies, click chemistry is a process that uses simple rules to easily click molecules into place. DNA-like materials could be shaped and optimized by an evolutionary process of variation and selection, Romsberg said. Let's say that you began with a material that had something and you wanted to change it or impart it with new function, he said. What we're selecting for is a certain property of the nanomaterial. And then, when we recover that nanomaterial, it presumably would have a very low quantity so you couldn't even analyze it. But then we could PCR amplify it because we have a polymerase that can work with these modified nucleotides, and one begins to think about what you could do if you could apply evolution to nanomaterials. Could we evolve new nanomaterials that do interesting things with the whole suite of improvements, stability, control, ease of duplication and improved ability to grab onto other molecules? The modified DNA is ready for commercialization, Romsberg said. He's talking with several companies who are interested in licensing the technology from TSRI. I think this will play out over the next few months, he said. I think it's very likely that this is going to be licensed and used commercially, which is something that's really exciting. The research was funded by the Defense Advanced Research Projects Agency, or DARPA. Related article. Scripps scientists get closer to artificial life with synthetic proteins. New DNA code makes synthetic protein Synthorx raises $10 million to make artificial proteins life engineered with expanded DNA code. Caption at 9:52 a.m. Pacific. OSIRIS-REx, short for Origins, Spectral Interpretation, Resource Identification, and Secure Regolith Explorer hits its closest point to Earth, 11,000 miles above Antarctica. September 22, 2017. Caption Bradley.Fikes at SudUnionTribune.com 619 2,931,020